Welcome, everybody. It's a nice crowd here to learn about sedges from Scott Ward. Scott is a master's degree from the State University of New York at Brockport, and he's been a research assistant in the plant ecology program since August of 2019. Scott's worked on a variety of demographic and inventory projects. His true love seems to be grasses and sedges, and so much so that we began to refer to him as Andrew, as in Andrew Pogon. But he's not going to talk about grasses today. Scott's going to talk about sedge diversity and identification in Florida. Scott?
All righty. Hello, everyone. As Eric mentioned, my name is Scott Ward, and I added this little line at the bottom from the perspective of a field botanist because knowledge of sedges is most appropriate when you're out in the field. I'm not necessarily a systematist, but I do understand a little bit of the taxonomy behind some of these groups, and it really comes in handy when you're performing field work and IDing plants. So a quick outline, I'll just introduce sedges, their habitats. I'll go over some patterns of diversity, talk about some basic concepts in their morphology, and then at least a few common terms that I'll probably use intermittently throughout, terms that are really valuable if you get into this group of plants. Although at first it can be a little overwhelming. And then I'll get into some examples of genera, basically a plural genus, right? And then I'll discuss some pertinent information related to some of these species and their habitats, their ecology, and then some morphological traits that I think really help when you get down to learning these genera. At the end, I'll get perhaps a little too in-depth to two of my favorite groups, Calcareous and Rincospora, using lots of pictures, species examples, and I'll try to be really specific about certain morphological traits so that you're clear what I'm referring to. So sedges are ecologically ubiquitous. They're virtually everywhere, in North America especially. Here's a picture of kind of the margins of a wet depression area in peninsular Florida. And if you were to get a little bit closer down to the ground, you'd notice that one of the dominants here is Rincospora pusilla. Even in upland habitats, this is also in the peninsula. This is more central Florida and Polk County. Really nice habitat of sandhill, where even though it's dominated by wiregrass in the ground layer, if you get down and really start looking at the diversity, you'll certainly pick up quite a few species of sedges, especially things like Rincospora and maybe even Scleria. Here's a Calcareous Prairie from Central Florida as well. Just incredible diversity in some of these grassland habitats. You could find countless species, especially of sedges, in these graminoid dominated areas. Kind of a wetland savanna also in Central Florida, where at Archbold we do a lot of our work. And if you start poking on in areas like this, you'd notice some areas are dominated by Scleria muhlenbergii, among other sedge species. Here's a recently burned kind of wet pine savanna with Rincospora latifolia. So this picture, in addition to all the previous ones, helps to really show that these are fire-prone, fire-adapted habitats. And even shortly after fire, you can see really robust responses in some of the species. And then even in habitats like in the Panhandle and riparian areas where they're not really prone to frequent fire return intervals, you can still see a fair amount of sedge diversity if you look close enough. And here on the right, you can see a picture of Carex grayi, kind of at its southernmost limit in North America. Even in habitats that we typically go for, we typically botanize in, not for sedges, it's hard to avoid them. Here's a pitcher plant bog or seepage slope. Some of these habitats are common in the Panhandle, really wonderful areas to explore. But even when you're in some of these areas, it's hard to avoid, genera like Rincospora and even some Carex species as well. Just to kind of reiterate this introduction, so Cyperaceae is the formal name for the sedge family. There's approximately 5,000 species worldwide, so it's quite diverse. In fact, it's the 5th most diverse plant genus worldwide. There's about 100 genera, give or take, in Cyperaceae. Like I said, they're ecologically ubiquitous and definitely fire adapted in many cases, and adapted to both wet and dry habitats. Up further north in tundra areas, boreal areas, you get a lot of diversity in the genus Carex. And then as you work your way down south a little bit more into tropical areas, especially savanna-like habitats and grasslands, you'll see a lot of diversity in general, like Rincospora. In North America, there is just under 1,000 species, about 27 genera according to the Florida North America treatment. And then in Florida, we have just under 300 taxa, give or take, depending upon the treatment, about 17 genera. And then compared to Poaceae, the grass family, which has quite a bit more taxa, but a lot more genera. So in my mind, Cyperaceae is a little bit more palatable because you have a lot less genera to learn, even though some of those genera like Rincospora and Carex have many species. So if you're botanizing or ecologizing in Florida, it's really essential that you know some sedges. I'm going to just kind of go over this real quick. This is kind of a, simplified version of basic graminoid or grass-like plants in their anatomy. Grasses differ a little bit in that they contain these spikelets, usually with a pair of glumes at the bottom. And then atop those glumes, there are one-to-many florets. And then within these florets, they consist of a lemma, palea, and other structures. So these are really characteristic of grasses. Moving on to rushes, graminoid plants, but not necessarily sedges, right? They have flowers with these this conspicuous tepal layer that kind of looks like petals a little bit, but it's a little different. And the fruits that are produced are capsules. The stems are round in Juncaceae, and some stems are round in Cyperaceae, but for the most part, most stems in sedges are three-angled. So sedges can be annual perennial herbs, usually, like I said, three-angled or trigonous. I'll use that term throughout. And then the leaves are usually three-ranked, and you'll kind of see that example, an example of that in a second here. Some species are very tall. Some Rincospora can get very tall, especially in tropical areas. But most species are about 1 meter or shorter. The inflorescence is basically the total flowering area of a stem or a culm. Consists of spikelets with bracts, which we also call scales sometimes. And these can be spirally or distichously arranged, meaning two-ranked on the side of one axis. And they're typically below or subtending the floral area. So subtending just means below. Flowers are mostly bisexual, but unisexual most often in Carex. With Rincospora, and you'll see towards the end of this presentation, I get a lot more into this, they contain these things called perianth bristles surrounding the fruits, which are achenes. And these are really crucial for identification, and you'll kind of see some examples of that later. Whereas Carex is really unique in that it contains these inflated sacs around the seeds called perigynia. And those are also rather diagnostic when identifying some species. So, there are, books upon books that have endless botanical terms. There's so much diversity in the plant world that, botanists use a suite of terms to try to categorize different morphologies. There's no way I could get into all these today, but there are a few that I think are really pertinent to this group in particular. So when I say annual versus perennial, I mean, are these plants completing their life cycle in 1 year, or do they have, root structures that help them to come back after, let's say, fire or a season? Are roots cespitose or rhizomatous? So here's a picture right here. Here's a cespitose example of a sedge, and then here's a rhizomatous sedge. Are the stems trigonous, so three-angled, or are they rounded, also called terete? Is the scale arrangement of the floral area distichous or imbricate, kind of spirally or two-ranked arranged? Proximal versus distal, that just means which structure is closer or further away from the ground. Scabrous versus glabrous means rough or smooth, and there's a lot of in between as well. And then when we get into certain characters like bristles, it's really important that you understand what the difference between retroversely barbed and anteversely barbed, what that difference is. And that just basically means, do the serrations face upward or downward towards the base or the top of the achene? There's a lot of other terms that are really important with this family, including scales, achenes, perigynia, spikelets, inflorescences. And I'll try to get into some of those examples with pictures later. So as I mentioned, there's a fair amount of genera in Florida within the Cyperaceae family. Here's just a list of those genera according to Warner Hansen's atlas. But there are a handful of genera that only have one species represented. For instance, Dolichium arundinaceum, three-way sedge, you can see on the right here is a map showing how widespread this species is, whereas another one, Genus nigricans, has a really interesting range. I mean, it's kind of, not really in between a lot of these areas, and a lot of the questions that biogeographers like to think about, why things occur here and not elsewhere. And there's just a few pictures showing these examples. Here's Genus nigricans here, taken from a glade in the Panhandle. You can also find this in Southwest Florida in wet calcareous areas. Bulbastylus robustus, this is kind of a strictly coastal species. This occurs in salt marshes and similar, tidal marsh influenced areas. And then Dolichium, really, even, they do have interesting achenes with bristles, but if you're just in certain boggy areas and you see leaves that look this distinctly 3-ranked, it's most likely going to be Dolichium. It's a really interesting species, and this 3-ranked character is really distinct. So I'm going to get into a few genera, and then I'm going to really focus in on Carex and Rincospora. So I'm going to start out with Bulbastylus. There's about 5 or 6 taxa, and these numbers vary depending upon the flora or the treatment that's used to categorize this diversity. Within this genus, there are Sesatos annual and perennial herbs. The inflorescences typically occur as terminal, so at the end of scapes or stems, flowering stems. And the spikelets, these little individual units here, can be either cylindric to ovoid, pedicellate, or sessile. And pedicellate just means borne on a small stem versus sessile, where it's basically attached right to the stem. And then the achenes are the little seeds, and they're trigonous, so they're 3-angled, and they can be very small to just somewhat small, and you'll see a picture in a second. The habitats are mostly these dry, sandy upland areas and similar disturbed sandy areas like fire lanes. There's just a few pictures. Here's a common one, Bulbastylus warei, especially in scrubby habitats in central Florida, has these really cylindric spikelets here that are really distinct. And then not necessarily all species are this ovoid like this. Sometimes Ciliatifolia or Coarctatus have these more slender spikelets at the terminal portions of the scapes. But even some species like Bulbastylus barbata are actually quite weedy. This one's a non-native, and this picture was just taken in downtown St. Petersburg. So sometimes they can just grow up out of, disturbed little cracks in the sidewalk. And, they're not super diverse, but when you start looking into characters like the achenes, you'll see in Bulbastylus warei, for instance, that at the apex, they have this really distinct sort of depression at the top. And some of these characters are really diagnostic when you start breaking these things down. Cladium is a really cool genus, not super diverse. There's only 2 species you'll find in Florida. The predominant one is Cladium jamaicense, sawgrass, and this species can get really tall, sometimes, upwards of 3 meters or even taller. It has really serrate leaf margins. So you can see this picture here. These serrations are just visible to the naked eye. If you're not careful and you brush past it, you can kind of cut your skin a little bit or hands. But it is the dominant species of the Everglades, and it's really important to remember that the so-called river of grass, the nickname for the Everglades is actually a river of sedge. Here's a picture of the inflorescence that can get quite tall, kind of taken at eye level, so kind of looking up. And then Cladium marisicoides is a much more widespread species, kind of found in a variety of calcareous type wetland habitats throughout the eastern United States, but you can find it in the Panhandle of Florida. So moving on to Siparis, this is one of the more diverse genera within the Cyperaceae. There's approximately a little less than 70 taxa in Florida. Really, just a little more than half of those are native. So there's a lot of weedy sort of rural species that are non-native as well. They're found in a variety of habitats. There's lots of species in these wet disturbed areas, for instance, but there are some species that are a little bit more specialized in their habitat. So when we start thinking about the morphology of these species, it's really important that you understand some of the terms and what they refer to. So starting out just looking at the whole plant, you'd see the cluster of all these spikelets is called a spike. And then when you start looking at these individual units, they're going to consist of these spikelets, and each one of these is a scale going up. When you break down that scale and you open it up, you'll find an achene. And whether this achene is 3-sided, like in this case, or 2-sided, kind of more lenticular or biconvex, That's really important when you go to start breaking down the diversity and IDing some of these things. So it's important to remember the scale, spikes, all the way down to achenes, also called a nutlet, and then think about the shape of these spike clusters. So digitate, kind of hand-shaped versus slender spikes. So there's a lot of really important characters to learn in this family, but over time they start to stick. Just looking at genus diversity of Cyparis, this is darker colors represent counties that have had more diverse collections of Cyparis. So you can see in the southeastern coastal plain, it can be quite diverse, but throughout the Northeast and Midwest, there's also a handful of species as well. Here's just a few examples showing these type of spike clusters and their shapes. So some are a little bit more globose and a little cylindric. Some are kind of more digitate. Here's more ovate, kind of the picture I showed in the previous slide. So there's lots of different varieties. Some characters, like in Cyparis haspin, are really important, like if the stem is really strong or if it's spongy in the case of haspin. So there's lots of other characters, but it often comes down to these spike clusters and then breaking down the spikelets. Here are a few more pictures just illustrating the diversity in Florida. Once again, there are some interesting characters, like if the culms have these septi, these little dividers in the stems going up, like in Cyparis articulatus. Some of them have these really these spikelets with really strong that are really strongly reflects, like Cyparis plugnettii. And then many species have these distichously arranged spikelets where it's sort of two ranked on this axis, the rachilla. And then some species, though, a few of them are spirally imbricate. So you can see the scales here kind of whirl around the center as opposed to just two-dimensional here. So once again, lots of diversity, lots of non-native, but also a fair amount of native species as well. Moving on to Eliacuras, the terminal inflorescences of this genus in particular are really unique in that they usually consist of a single unbranched spike with a terminal inflorescence, like in Eliacuras tuberculosa here. So some spread vegetatively, and others reproduce, using reproductive means like producing spikes with seeds. It's important to think about the size of these spikes and how they relate to the culm below. So some species have spikes that are much wider than this culm, whereas others, you'll see next slide, have spikes that are about the same width as the culm. And the achene characters are also really important. And I'll get more into achene morphology later in Rincospora, but you can just see some of these, the difference between these two species are pretty distinct. Eliacuras interstincta, this is a really common species, often colonizing these sort of man-made ponds along the margin. But you can see what I was talking about here. The spike width at the top is about the same size as the culm width, so it's not much wider. Bulbastylus, there's some non-natives and natives as well, but there's overall about 9 to 12 taxa in Florida. It's important to think about, once you break down those reproductive structures again, if the achenes, those little seeds, are lens-shaped, lenticular, or trigonous, three-angled, is the plant an annual or perennial? You can usually find this out pretty easily just by sort of kind of lightly tugging at the roots and seeing if the roots are really robust, likely an adaptation to perenniating, or are they really kind of easy to pull up, established by seeds, reproduce, and then die. And then also looking at when you break down perennial species, if they're Sesatos or rhizomatous, meaning tufted or kind of tufted or long rhizomes at the base, and are plants really tall? Are they pretty small, like in the case of Bulbastylus autumnalis? Moving on to Fiorina, a small handful of taxa in Florida. Also important to think about if these taxa are rhizomatous or not, like in the case of Fiorina scarpoidea, which has these really long rhizomes where culms kind of shoot up at intermittent points. Spikelets are often in terminal sessile clusters, like you can see here. And bristle length can also be pretty important when you break down the achenes. Moving on to Chenoplectus. This is a really cool genus, and species within this genus can have either triangular or terete, so rounded stems. There's about 7 taxa in Florida, and they usually have 1, maybe 2 foliaceous involucral bracts. This is what this refers to right here. And in this genus, it can often look like a continuation of the stem, but it's actually not a true continuation. It's its own kind of separate adaptation or separate bract. So, and then lastly, it's important to think about if these spikelets are sessile or in this case stalked. So if these spikelets are attached basically right at the culm right here, like in Chenoplectus pungens, or if they're pedicellate like Chenoplectus tuberculatus in this case. Here's just a few more pictures showing two species. Most people are probably familiar with Chenoplectus taberni-montani, that's soft stem bulrush. Specimens in Florida, I think, get really, really large. I've seen some stems that just, are really wide and kind of full of these spongy parenchyma. And then, akene characters can also be kind of important once you break down reproductive portions of the plant. Chenoplectus tuberculatus is probably not very common in the peninsula, a little bit more common as you get into the panhandle. Moving on to Scleria, the nut rushes. These are really interesting and sometimes kind of quite small species. There's about 13 to 20 taxa depending upon the floral treatment. Looking at this blue circle here, this is the whole inflorescence, or if it's fruiting, the infructescence. And then you can kind of break it down into these clusters. And then within these clusters, there are spikelets. And then if you kind of peel out the akene here, you can actually get to sort of the lower reproductive structures. And with this genus in particular, it's really important to consider if the akenes come with this structure called a hypogonium or not, sitting at the base of the akene. Here's the tip, so this would be the upper part and then the lower part. And then if it does contain a hypogonium, how many lobes, what kind of shape is it in? So there's lots of different ways you can take these keys just due to the diversity. And then thinking about these spikelets, if they're drooping or erect is also pretty important. And then the texture of this akene really comes into play too. Here are just some examples of species you can find in Florida. And as I mentioned here, there's kind of a complex of Scleria ciliata. There's a few varieties recognized by certain authors. And you can just see the surface of this akene almost kind of looks like a pitted golf ball. And there's other variations within this complex. And then other species are kind of smooth, like Scleria elegantha. Some species just look really interesting overall, like Scleria distans. And then some spikelets are kind of more pedicellate and drooping, like in Scleria muhlenbergii, which is a pretty common component of wetland communities, at least in Central Florida. Okay, so getting away from kind of the rest of the genera, I'm going to hone in a little bit more on Carex and Rincospora since they're so diverse, especially in Florida. So Carex, true sedges, Carex are distinct from other Cyperaceae due to the presence of a perigynia, so this inflated sac right here. If you open up these little perigynia here, you can find akenes. And they're often trigonous, kind of sharply three-angled like this, but sometimes they can come in other forms. There's about 75 taxa in Florida, but there's about 2,000 species worldwide. So, it's a really, really diverse genus. There's many ways to learn this genus, but, some species are even so distinct that you can look at it and say, okay, I'm pretty sure, based upon where I'm at, I'm pretty confident I know what that species is because, nothing else looks like that. Whereas other species exist as part of these really diverse, morphologically similar groups that we call sections. Some sections are even really diverse too, like the notorious ovalis group, for instance. So there's still some phylogenetic work to be done on kind of sorting these sections out. But to me, it's a really useful skill to learn some of these sections because you're taking, let's say, 75 taxa in Florida. Oh, okay, we know there's only 26 sections. So it really cuts down on the possibility once you know kind of a little bit about these groups. As I mentioned earlier, Carex is much more diverse as you get into temperate, kind of cooler areas, especially, these radiations in forests. But Carex is still pretty diverse in the southeastern United States, even with some localized endemics, so plants that are found in some areas and nowhere else. You know, to really get into Carex ID, it's important to kind of be familiarized with certain anatomical traits. So, you know, even basic things like knowing, you know, what a spike is and what, you know, Perigynia is, achenes is really important. Some of these kind of splits, when you think about how they're organized in keys, come down to things as basic as, you know, are the styles in numbers of 2 or 3? Is the achene shaped like a lens, or is it more sharply 3-angled? Are the spikelets kind of sessile on the combs? Are they borne on these little stems coming off the main stem? You know, do culms have only male flowers, or do they have both male and female spikes? And then, you know, there's different terms for the arrangement of the spikes along the culm. And then also, once you get down into the sections, which can take, you know, a little while to learn some of these sections, but once you get into it, you learn that, you know, subtle characters like differences in the Perigynia shape and even bract characters can also be really important. So here's just a picture of Carex striata, and the white arrow points to the styles. So that's in numbers of 3 in that case. This would be in subgenus Carex, which I'll talk about in a second. The blue arrow points, if you can look real closely there, the blue arrow points to the scale that subtends the Perigynia, which is circled in yellow. And then the yellow arrow points to the bract, in this case, longer than the inflorescence. So all these are really pertinent when you start to think about how to key these things out. I'm not going to go into this too crazy, but with such a large genus, you know, there's been a lot of work on splitting them up into subgenera. All you really need to know is that there are a few key characters that separate the 2 main subgenera, Vignia and Carex. You know, spikes that sessile bisexual spikes kind of here. In this case, you know, the female spikes are above the female portions above the male. And then, you know, stigmas in numbers of 2. Achenes are also lenticular with this group of plants. Whereas most Carex fall into this main group, Carex, and they're a little bit more dynamic. They usually have pedunculed spikes that are unisexual. And that just means, you know, this is, for instance, right here, this is the male spike and this is the female spike, and they're borne off separate stems. And stigmas are in numbers of 3. So when you start to break down the floral morphology a little bit more. And then also, you know, achenes can be trigonous or terete, but are often sharply trigonous in some cases. Okay, so as I mentioned, there's terms for when, you know, male spikes occur above female spikes and vice versa. So on the left here, this would be an example of an androgynous plant in Carex flavo, where the staminate spike sits above the female portions. This is Carex buxbaumii, one of my favorite species, where the female portion with the Perigynia sits above the male flowering portions right here. So this would be gynecandrous. And then even within this species, it can vary quite a bit in that sometimes you'll find it and it'll be androgynecandrous. So there'll actually be male portions above and below the female portions in the distalmost spikes, and you'll see that in a second. So these entire area, this whole thing would be called the inflorescence or infructescence in some groups if they've gone to fruit. Here's an example of a terminal male spike, just meaning it occurs at the uppermost portion of the culm. Here would be a spike that occurs laterally, and this would be more proximal in this case, meaning it occurs lower to the ground. So you'll see distal versus proximal a lot as well, and that's what that refers to. There's other characters that are really important, especially when you get into length, color, shape. For instance, the carpellate scale subtends the Perigynia. So, you know, each Perigynia has a scale that sits right below it. There's characters like bracts that are really important to know how long are they? Are they longer than the inflorescence, or are they shorter than the inflorescence in some cases? Of course, I mentioned the Perigynia that houses the achene within. And then even, you know, staminate portions, even though they don't look kind of more swollen, like, you know, because there's no Perigynia, they're still kind of important and can be quite colorful in some cases. So staminate portions have scales as well. So Carex is, as I mentioned, much more diverse as you get into these cooler temperate climates. You can see in the Northeast here, those dark green areas represent counties with really high Carex diversity. But as you get into Florida, if you're in the Panhandle in particular, there's actually a fair amount of diversity. The record holder, at least according to the Florida Plant Atlas, is Gadsden County, and that has at least 58 taxa vouchered. And actually, that should be 59 because there was a new Carex found in there recently. So there's always room to add more taxa to these lists. They're sometimes incomplete, but about 60 taxa is pretty diverse for one county. So as I mentioned, Carex is broken up into sections. I'm not going to get too crazy into this, but I'm going to show you some examples of a few species within different sections. Ovalis takes the cake, as it often does. This group is just really diverse within Carex. There's only 8 species, though, in Florida. So compared to maybe if you were in New York, where you get almost 25. So it can be, it's a lot more palatable down in Florida. But there's other sections that are pretty diverse as well, like the Grizzly, for example. I'm not going to get crazy into this, but I just wanted to illustrate some of the diversity and the different morphologies based upon these different sections. So like the Stellulati, for instance, the star sedges, they kind of look like this, subgenus Vignia. They have much more lenticular shaped Perigynia versus, let's say, something in Folliculati, which could have really long, thin, slender Perigynia, or in some cases, like Carex turgescens, might have a little bit more swollen or turgid Perigynia. And then when you get into sections like Lupulini, I mean, there's really, no mistaking some of these species. These are the Hopsedges, called that for a reason because they kind of resemble hops, the Perigynia. So species like Grayi, Intumescens, Lupuliformis, can be common in the right kind of habitats. Grayi is a little bit disjunct in the Panhandle. It's at its southern limit, but Lupuliformis kind of takes over where Lupulina would be further north. So it's important to, understand then characters like the achene. The achenes in species like Carex gigantea are sharply trigonous. Some sections like Hymenoclani, also called the green or woodland danglers, have these pedicellate drooping spikes that are really characteristic. And then 2 species you can find co-occurring actually, like in the Panhandle, are Carex stabilis and venusta. I'm going to just go over these next two slides really quick, not to bog anyone down, but you can just see the diversity of these sections. I mean, they're really distinct looking. Something like Carex leptalia, which has Perigynia, here's my thumb, for instance, for scale, really tiny spikes compared to maybe Carex verrucosa, which has these really large, kind of showy, glaucous blue female spikes. So, it can vary quite a bit. Sometimes Perigynia are really interesting, like the narrowly obconic Perigynia of Carex frankia, kind of a more southern species. So some of these, characters begin to stick when you associate them with, oh, this species looks like this, and I know that I can associate this term with it now because, very little other species are like that. Just a few more sections. Here's Carex vexans, which is in the Ovalis section. Really not super diverse in Florida, like I mentioned, but can be very diverse as you get further north. Other sections like Palidosi, Laxiflori can be a little quite diverse in Florida. Some sections don't have very many species. So, it's not necessarily crucial to learn the sections sometimes because there's not many representatives like Carex dasycarpa. But other sections like Grizzly and Multiflori, it can really benefit to learn some of these sections because diversity can be quite high. Some characters not related even to, some characters, I shouldn't say, some characters are even important, are additionally important compared to some of the pictures I showed you earlier, such as beak characters. So at the terminal portion, at the apex of Perigynia, are these beaks. And in species like Carex camosa, the beaks are strongly outcurved, whereas in the same section, Vesicari, Carex lurida, has these beaks that are much more straight and a little bit shorter. So even things, just subtle beak characters like that on the Perigynia can sometimes, boom, get you from one species to another. If let's say you're out collecting or doing field work or inventorying somewhere, would like to know what Carex species are there. Some of these characters can get you there within seconds, really, when you see them. Here's just an illustration from Florida, North America, showing different species in Carex ovalis. Most botanists often look at, they find an ovalis in the field and kind of have this kind of reaction. But when you start to break down, the specific paragenia characters, and spike characters and really minute measurement differences, you start to learn sections like this more. This is still one that I need to spend a lot of time with, but fortunately, like I said, in Florida, there's not too many representatives, so it's a little bit easier to grasp them down here. Okay, so moving on now to Rincospora. This is a really fun genus to get into if you're doing field work in the South, especially in Florida. There's about 250 taxa or so worldwide, and about 67 taxa in Florida, give or take the taxonomic treatment. Rincospora overall has a greater affinity for warmer subtropical areas versus the temperate affinity that Calcareous have for further north. And field ID honestly can be quite difficult compared to Carex, and that's because of these small Achene characters that I'll talk about in a second. So things like magnification are quite crucial when you get into learning some of these groups, especially when you break down spikelet morphology and Achene shapes. But Rincospora is found in a variety of habitats, a little bit fewer in uplands, but you can still find a handful in upland habitat, and then really high diversity in wetlands. Okay, so just to show basic diversity patterns of Rincospora in North America, thank God for BONAP because I use it all the time. You can see in the Panhandle especially, and really just throughout the Southeast coastal plain, Rincospora diversity is really high. You go up to New York, you might get, I don't know, 7 species or so, I think, if I remember correctly. So, whereas in Florida, you have 67 at least. So it's definitely much more tropical kind of genus. And of course, as you get into Central and South America, you find lots of species as well. The record holder, at least according to the atlas, the Florida Plant Atlas, is Bay County with 45 taxa. But overall, the Panhandle is a really good spot to be if you're into both Carex and Rincospora. Here's just a side-by-side picture that I really like taking these pictures sometimes because it helps to illustrate basic differences between different species. You can see, like on the left here, Rincospora tracea has these globose or round spikelet clusters, whereas other species have these showy involucral bracts, like Rincospora latifolia, and then groups, like that I'll talk about in a second. Some spikelets are very long, tubercles are very long. So overall, many species often tend to look like this, where they have these sort of, nondescript brown spikelets that are in these fascicles. But when you really start to break them down and tease apart the achenes, you can start to tell the differences. So as I mentioned, Rincospora can be quite variable in the overall size, shape, color, the habitat preferences, the height of the culms or the stems, and then of course, what I'll finish with today, the achene characters, which are really crucial. But you can just see some of the differences here, like in Rincospora macro with white spikelet or with white scales compared to most others that have these brown scales surrounding the spikelets. Some species have really tiny inflorescences like Rincospora pusilla and subsequent achenes. You really benefit from using things like a basic cheap dissecting microscope you can get online or whatever. So this orangish sort of yellow circle refers to the whole inflorescence or infructescence if the culm has gone to fruit or seed. Basically the sum of the reproductive parts. And then subtending these spikelets, the spikelet clusters in some species are these really showy, well, really just a few species have these really showy involucral bracts. Oftentimes they just kind of look like continuations of the stem in some cases with lateral inflorescences. So sometimes they're not very, actually often they're not very showy. Spikelets occur in these spikelet clusters. So, you know, if you were to tease this apart and really get down to it, you'd find an individual spikelet that consists of, you know, sometimes one or maybe just a few achenes. And I'll show a better picture of that in a second. And then when you break down that reproductive structure, that's when you start to really get into scale characters and then achene morphology. Here's an illustration from the description of Rincospora marliniana, more recently described, member of the Plumosae. And this blue circle surrounds a spikelet. So really one spikelet, you know, this small unit, this small flowering unit is quite small when you think about the entire spikelet cluster, the entire inflorescence on a species. These characters are often really important and needed to tease apart, and you need to tease apart these spikelets from within these clusters to learn things like, you know, possible pedicel length and scale characters. Root characters can also be really important, so sometimes it's really crucial to collect below ground parts. Let's say if you're collecting and IDing. And then achenes are absolutely essential for proper, you know, and correct identification. Achenes are nested within these often brown colored scales, nestled within spikelets. And it's really easy to just, you know, take the achene within the scale and just kind of squeeze it out into your hand and look at it with a hand lens or put it on your dissecting scope, either way. Okay, I'm just going to do 4 quick examples of different achenes. So the black arrow here points to the tubercle in Rincospora Traceae. So you can see here, this tubercle is actually quite long. It's rather slender. It's within this group that has these really long, conspicuous tubercles. So this is something you could look at just, you know, without even a hand lens and say, oh yeah, that tubercle is quite massive. Whereas you start looking at characters like the perianth bristles right here. The length of these is really important. Did these bristles, for instance, exceed the achene and the tubercle? Does it exceed the whole thing? Sometimes you have to be careful in certain keys and figure out, you know, if they specify whether the total measurement of the achene includes the tubercle as well, or sometimes it just includes the achene body and then the tubercle measurement is separate, like in Weekly's flora, for instance. The direction of these serrations on the side of the bristles are really important as well. Basically, the barbed direction. So in this case, it's kind of hard to see, but they're facing upward, so they'd be antrorously barbed. And then if they're facing downward, you'll see in a second, they'd be retrorously barbed. Like in this one. So I think this was Rincospora cephalantha. You can see this tubercle is still pretty long, but not quite as long as the last species. But the barbs here are facing downwards. They're facing the base of the achene, the more proximal portion, right? So this is going to be in a small group that has retrorously barbed bristles. Rincospora wrightiana, this is a neat one because, you know, it's within, this species has a really uniquely shaped tubercle. So sometimes tubercles can have uniquely shaped forms, which can really help with identification. Sometimes they can have kind of color differences in the center of the achene versus the outside, you know, sort of rim of the achene. But once again, you can just see how much shorter this tubercle is. The achene's much shorter, and the bristles don't exceed even the body of the achene in this case. And then to review, these barbs are antrorously or upward facing from the base of the achene. And then lastly, some achenes don't have bristles at all and are sort of nondescript, like in Rincospora chapmanii. So sometimes the tubercle is kind of this sort of plain deltoid look. Some color differences, like if you want to separate Brachychaeta from chapmanii, for instance, there's a little difference there. So, you know, it depends on the treatment, but also the subtleties of that treatment and sort of what the authors recognize as the differences. So you can just see from those 4 examples, achenes can differ quite a bit. I'm just going to go through each of the main 5 groups that are represented. They're kind of broken down in Weekly's flora. I really like the way he organizes the achenes into groups like this, and other floras like Wonderland and Hansen as well. Their 3rd edition basically follows the same format. So I really like this, and you know, if I were to promote a way to learn the groups in Florida, I would use this as I will. So the first group is Haploid Stylee. They have simple styles and really long tubercles. So these two species right here, you can see they're quite large. Here's a picture taken a couple years back. This is the culm of, can't remember if it was Rincospora inundata at the time, but one of the members of this group, and you can see it's almost as tall as me. And of course, there are species like in more tropical areas that are even taller, but generally quite robust, pretty tall. A little shorter in Rincospora Traceae, which is in a different section, but in that same large group. And then these long tubercles, and here's a few examples. You can just see how long these tubercles are. Characters like bristle length, like if there's alternating rows of bristles that are shorter and longer, might be diagnostic to get down to species. And then overall bristle length is important. With this group too, it's really important to collect rhizomes because they can be diagnostic as well. So I kind of went over this already, but there's 5 species in Florida. Rincospora Traceae is the one that has globose or round clusters. And the rest kind of differ in having these more cylindric, longer, thinner spikelets. As I mentioned, they can be quite tall. Rhizome characters are important, and it's important to think about if the bristles exceed the length of the achene or not. Take a sip of water real quick. Okay, the second group I'll talk about is Dichromina. These are, it's the first group I ever learned years ago when I took my first ecological trip to Florida. I mean, it's just hard to miss it. It almost looks like a regular flowering plant, and then you realize it's actually a sedge. It has these white foliaceous bracts. There's only 3 species in Florida, and really only 2, kind of throughout. Floradensis is more restricted to South Florida, and that's a Sesatos species as opposed to Rhizomatous and the 2 other species, Colorata and Latifolia. Like I said, they have these really showy bracts, so they're hard to miss. And the number of bracts is how you separate Colorata from Latifolia. The achenes, they're important. They're not often looked at with these species because you can usually just look at the bract number and ID them, but I still think the achenes look pretty cool. So the beaks are a little bit longer and less deltoid shaped in Latifolia than Colorata. Okay, moving on to really in my mind, the most showy of the achene groups, the Plumosae. This is a really cool group and found in a variety of habitats, everything from basically scrubby flatwoods to seepage slopes. It's really, it covers the gamut of habitat types. You can see here in this picture of Rincosphere Megaplumosa, a relatively newly described species. I mean, not super recent, but wasn't known about for a while. But you can see here, the bristles are actually so long that they protrude from the actual scales. And here you can actually see the tubercle and the achene below. If you look at this achene closer up, you can just see there's no mistaking the species. Basically restricted to like 4 counties in peninsular Florida in these scrubby kind of habitats. So it's, a localized endemic, but really neat looking, especially compared to the other Plumosae. When you start looking at the other Plumosae, you're sort of looking for things like differences in the bristle characters. So like Marliniana, which was also more recently described, is really more plumose at the base and not throughout, whereas Pineticola and Plumosa are plumose throughout. And then it's important to think about like constriction. So in Oligantha, at the tip of the achene body, there's kind of a constriction, or I'm sorry, for Galliana. And then the bristle length in Galliana in particular is quite short compared to other species. So, achene constrictions, even tubercle length and achene length can really be really important, like when you're separating Pineticola or also called intermediate from Rincospora plumosa. But otherwise, these can be quite tiny in certain habitats, and so you might even miss them in certain cases if you're not looking carefully. Moving on to this next section, what's really distinct about this group is that the bristles are retrorsely barbed. So you can see here, just to review, the barbs here are facing downward. They're facing the base of the achene versus facing upward or antrorsely barbed. There's only 6 species in Florida that are within this group. So it's generally not an overwhelming group to learn. Let's say you go out, you collect some Rincosporas that don't really look that descript, bring it home, you look in the dissecting scope, and you see that, you know, these barbs are facing downwards. Boom, you already know you're within that section, and you only have, you know, 6 species to choose from. So once again, like the Plumosae, there's just subtle achene characters that can, you know, get you almost immediately to, you know, a much smaller section instead of that sort of ominous, you know, 70 species or so that you started out with. Some characters are really similar, like Microcephala and Cephalantha, and some characters, you know, related to the length of these achenes can be really crucial when separating them out. Some species have tons of barb or bristles, like in Rincospora macra, kind of the southern version of Rincospora alba with these white bracts. So outside of the achene, there's other characters that can be important as well. The last group, which I sort of just call all others, includes many different sections, like true sections of Rincospora. But I think of it this way as either being bristles being antrorsely barbed or bristles just being absent altogether, as kind of being this almost catch-all group at the end. So, and then once you get into, let's say, if bristles are antrorsely barbed or if they're not present at all, then you have to get into characters related to the achene surface, whether the achenes are smooth, like in Chapmanii, or if they're kind of honeycomb reticulate or ridged, which I'll show a better example of in the next slide, like in section globularis. So for me, some of the characters, you know, some of the terms are rather synonymous. So some achenes, like some species like Rincospora eximia, have these sort of transversely ridged achene surfaces, which I think in some cases tends to be less wavy than what you get in a species that might be considered wavy rugose. So once again, a ton of terms that botanists have for these really minor differences. But when you're looking at these things in the microscope and you're keying them out, it's really important to know that those subtle differences can be crucial in figuring out whether you have, let's say, a species that's really common and kind of weedy, or a species that's potentially very rare and more limited in its distribution, like Stenophila, for instance. And then sometimes some of these patterns are really kind of cool looking, like in Rincospora pinetorum, recognized by some as a variety of globularis, as being kind of honeycomb-like. So, you know, in short, there's so much variety in achene textures and characters that it's really, when you start to look at these things closer up and you start to see how much differences there are under the scope, it's really kind of fascinating. Some differences can be very subtle, but as I mentioned, very important. Some characters like the length of the stipe beneath the achene, for instance, is really important in figuring out if you have connipes or, you know, so there's lots of really subtle characters as well that really come into play. And, you know, someone who's not really into botany very much might look at this and say, oh, well, you know, they all kind of look alike. But when you start to realize that not only do they look different when you get down to it, they have different habitat preferences, and, you know, there's different ecological requirements for each species. And each species can tell you something totally different about, let's say, the management or fire history of an area. So you know, to me, it's important to respect and understand this diversity because it tells you so much as an ecologist. Here's just another picture kind of showing species within this catch-all group. I mean, you have species like Rincospora grayi, which prefers, you know, upland habitats like sandhills. And you have species like Rincospora eximia, which is really not very common at all, only has a few collections in peninsular Florida and is really at its northern limit in Florida. Species like Rincospora ciliaris, which have these smooth achenes, but have these little cilia on the side of the leaves that are really diagnostic. Some, you know, for instance, or for example, here's one species, Rincospora microcephala, and here's 1 centimeter. So, you know, these can be quite tiny, but important when you're identifying and keying them out. Like I mentioned, some species are restricted in the range, like Stenophila. Rincospora compresses, a really cool species with almost kind of biconvex achenes that you can find in the panhandle. And then other species that just kind of look altogether different depending upon, you know, your knowledge of the genus. So it's really cool to see this diversity and kind of understand it and really work towards, you know, studying it. Maybe a quick bristle acronym you could remember for these 5 groups as kind of set forth in many keys. So tubercle length, are they super long and conspicuous? Can you see them without magnification? Are the bracts showy and white, like in, you know, Colorata and latifolia? Are the bristles, if present, are they plumose? If bristles are present and they're retrorsely barbed, that's a small group. And then antrorse or absent kind of represents this group that has antrorsely barbed bristles or no bristles whatsoever, so absent. I just came up with a, you know, a silly little acronym right here, tedious botanical pictures, relativize at Keen's always, but you can come up with whatever you want, but it helps to kind of think about how you can split up these really large genera into smaller groups. Here's just some more pictures, just because I just love this group and love studying it, and it's really cool to see the diversity. You know, this picture represents species that were all collected in one single management unit, and it was within the year of fire. So, you know, it just helps to show that not only is fire integral into some of these ecosystems, but diversity is just astounding. And this isn't even all the species that were seen that day. So here's just a kind of a ruler for scale to show you the difference in these achenes lined up. And then this is how I sort of group them on my dissecting scope at home, just kind of showing the size difference as well. So to kind of wrap things up, I know I went kind of deep into some of those genera, but you know, overall, it's important to know that the sedge family is very diverse, but genus diversity is not overwhelming. It's not like grasses where you have a lot of different genera with maybe only a few species. You know, there's a few genera that are very diverse, kind of in this order: Carex, Cyparis, Rincospora, and Eliacuras. But depending upon where you're at, your knowledge of the ecology, the habitat, and then your knowledge of, you know, minor morphological differences, you can start to hone in on this diversity a little bit better. As I mentioned, they're ecologically ubiquitous sedges. They can be found in all sorts of habitat types, in uplands and wetlands, high elevations, low elevations, Arctic, tropical areas. To really start to grasp this family, though, you have to really break down and collect and start to key these things out. Things like dissecting scopes, hand lens are absolutely crucial, because sometimes there are these really small features that are hard to see with the naked eye. I also, as you can tell, like to take a billion pictures, and that often helps to kind of picture in your head, okay, this term is called this, and now I actually have a picture of it. And, you know, now it's in my head forever that retrorsely barbed means this. So, you know, as a visual learner, it's really helped me to sort of learn some of the diversity. I also recommend taking an easy species and running that through the key. So take one that you already know, Colorata or latifolia, Rincospora colorata or latifolia, and then just run it through the key and just say, oh, okay, that's what this term means. That's what that refers to. And then often newer treatments, so species that are more recently described, have really fantastic illustrations within those papers as well. So you really have to maintain the knowledge of, you know, diversity by using different keys in floras. And, you know, no one learned 5,000 species in a day, although I can think of a few botanists that probably, you know, could learn it, you know, a similar number just because of their vast knowledge. Some resources I recommend, at least within Florida. So Alan Wakely just came out with his more recent iteration of his Flora of the Southeastern United States. You know, a lot of respect for this. There's just a huge area that it now covers, about 10,000 species or so. I mean, it's a massive undertaking when you think about these projects. But fortunately, he has floras that are adapted for each state and each area. So, you know, there's a key just for Florida, for instance. Another classic resource, Wonderland and Hanson's third edition. There's, I mean, the second edition and first are pretty good too, but the third is just has more updated taxonomy. You know, generally there's some taxonomic differences between these two resources, but that's what's great is you can use both and kind of compare and contrast and see, which species are recognized by some authors and which aren't. And then, some of these terms can be overwhelming as well. This is a great little, I think this was a Christmas gift I got years ago. So it's a nice little glossary guide to some of these morphological traits. I also use Flora North America, the online version, and the book quite often. Volume 23 includes the Cyperaceae, and this is just open source. They have a new beta up now too, so the website looks really kind of sharp. And you can see these really in-depth descriptions for different species, and they've had a suite of illustrators that do really fantastic close-up drawings of different species. So there's really a ton of resources available nowadays to learn some of these diverse groups. To kind of wrap up, I'd like to acknowledge first off Archibald Biological Station, in particular the plant ecology program, my boss Eric Menges, and my colleagues Stephanie Kuntz and Lexi Siegelbates, in particular, is for helping me with all the projects we do related to demography and other ecological projects. Also for their kind of understanding that sometimes when I'm maybe lagging behind in the field, it's because I'm staring at some tiny achene or something. So I also am really appreciative of their patience, and especially the interns' patience when we're out in the field, and kind of understanding that, passion is definitely encouraged at Archibald Biological Station. I'd also like to thank the Flora North America Association. I use the resource all the time. It's great that it's open source. These kind of resources are invaluable. Botanists, I mean, there's so many botanists and naturalists that I could go through, but in particular in Florida, here's just a list of some that either directly or indirectly have either enhanced my knowledge of Florida ecosystems and the species components, or have just helped me to really come up to speed with some of these diverse groups down here. I'd like to thank all of the various fire managers across Florida that create these really interesting fire adapted, that help to maintain these really interesting fire adapted communities, conservationists around, and then I'd like to thank all those that support Archibald. Here are some of the sources I've used. And with that, I will take any questions.
Thank you, Scott.
Hey, Eric.
There's several questions, but I wanted to ask one of my own first. You've shown some remarkable diversity in flowering structures and in achenes among the sedges. And so is there a comparable diversity in terms of pollination and seed dispersal, or could you summarize how sedges produce in terms of their pollination and their seed dispersal?
So yeah, a lot of graminoids are wind dispersed, or wind pollinated, I mean. But this group in particular on this picture, Dichromina, there actually have been some studies looking at pollinator visitors. So there have been some studies in the tropics and relatively newer research, although some of it dates back a little bit, looking at insect communities that actually use these more showy species. So for the the less showy kind of brown fascicled species, I couldn't speak on those. Maybe wind pollination is more important, but for the showier ones, there's probably some insect groups.
And for seed dispersal?
Oh, for seed dispersal, well, I mean, this is a field that sense of sedge ecology. I'm not super well versed in, but it's generally thought of that perigynia help with water dispersal because they're kind of inflated and help to float across different water areas with water. Whereas I think bristles with Rincospora really help to attach to, let's say, fur or something like that. So, I think there are subtle morphological traits that help to disperse like those two.
Okay, and Lara Bird asked, will invasive sedges do better and spread their ranges given climate change?
There's definitely some Siparis that I think will keep working their way up north. There's a lot of non-natives in Florida that are still just kind of in Florida. And in 20 years, maybe there'll be more of a problem in, let's say, Georgia or Alabama. So I generally would say, you know, I mean, with a general disclaimer that I'm not really a climate expert, is that, yeah, I think they're going to be more of a problem, especially in Cyparis. There's a lot of non-native kind of weedy ones and certain species that are kind of on the watch list, if you will.
Okay, and Hilary Swain asked about characteristics of the few sedges that are insect pollinated as compared to wind pollinated, and whether any of those insect pollinated ones are widespread in Florida.
Colorado is quite weedy and almost all over in ditches, you know, on roadsides. So perhaps perhaps being showy and attracting more pollinators actually helps it to disperse more. So I think there probably is a component there, but you know, some of these species that are really tiny and are, you know, maybe more wind pollinated, I think they're kind of restricted in the range for a reason. I think there's something to that. So.
And a related question from Toby Shaya is, why are so many sedges specialized in terms of their habitat preferences?
I'm sorry, could you repeat that one more time?
Why are so many sedges specialized in terms of their habitat preferences?
I mean, I think that might come down to this concept of niche, you know, niche partitioning. I think, you know, there's these plant communities, as beautiful as they look, they often can be quite competitive, although there's some symbioses. But I think, yeah, it's just an example of all the different habitats, and yeah, just the variety of habitats and open niches that are available in Florida. I mean, Megalocarpa grows in the driest of scrub, and so there's even these really harsh upland examples too.
And Paza asks, what's your favorite sedge so far that you found in your entire career?
In my vast career of, you know, not very long at all, I would say, you know, if you don't love Rincosphere Megaplumosa, I mean, there's something wrong. It's just the coolest species. But I also really like some of the really tiny ones. There's ones that I really like because they're hard to find, like Rincospora thornii. So, you know, there's different reasons, but Megaplumosa there's nothing like it at all.
Okay, I'm looking. There's quite a few entries in the chat, and I think there's some questions. That's what I'm looking for now.
Okay.
I'll start with one that a couple of people asked, Scott. Is there any chance that you would share your presentation slides with a PDF with the group?
That I would share with who? With
the group of attendees, or possibly with other groups.
Oh yeah, I should have put my email on this slide. It's sward@archbold/station.org. I could put it in the chat too, but you can email me and I can try to get you a copy.
All right.
And I think Archbold Expeditions puts up the presentations on the YouTube page as well, right?
That's correct.
We got a few more questions that have come in. David Austin asked, are sedges important bird food?
Oh man. You know, there's also, I don't know a ton about this, but I know waterfowl use, you know, let's say species of Persicaria a lot in these drawdown wetlands. And I'd be surprised if they didn't somehow use some sedges as well. I know grasses are really important, so yeah, maybe there's some potential. Although some of those, you know, achenes, they're probably not very digestible. So, yeah, they're really important for habitat integrity. The wildlife uses, I think, still need some work as far as, you know, what uses them.
Okay, and I have a specific question here. I hope I don't butcher the names, but Jackson Jablowski asked about the Cuban bulrush, Cyparis blepharoleptos, formerly Oxyocarion cubense. Are you familiar with this species and its distribution in Florida?
Yeah, that one actually occurs in Lake Annie. It kind of likes these floating vegetation mats along the edges of, you know, shallow lakes and things of that nature. I actually just saw this again last weekend in the Myakka River floodplain area along the banks of the Myakka River. So it's certainly around. I couldn't tell you the range right off the bat, but it's definitely a troublesome species, and particularly like in Lake Annie, it's just on these floating mats that have really cool native species, like Xyris corelliorum, but they kind of creep along in these mats, so it's hard to control. So I would say, and they form these really dense kind of root systems in those floating mats. So I definitely would say that one is one to keep an eye on.
I'm going to ask one more question, but I wanted to point out to the people here that Edwin Bridges has provided answers to 3 of these questions too, with additional information.
Definitely not surprised.
Anybody wants to find out more about their question, you can look in the chat. Let's we'll wrap it up by a question of why did you decide to become a botanist?
Well, I'm sure there's some silly comment I could come up with, but I think I was fascinated when I was younger in how plants grow and seeing the response of plants, to basic growth requirements. I mean, just seeing how plants grow really fascinated me. And then, some of these things can just be sort of inconsequential. Like I was in a program at FLCC in New York, and part of the degree requirement was to pass this test where you ID a bunch of trees outside, like 200 questions or something like that. And as soon as I started studying for that, I just haven't stopped. So that was when I was like 17, and you just can't stop, because once you catch that bug, you keep learning more and more, and then you realize that wow, now I know 100. If I keep working at it, I'll know 500 species and 1,000, and so there's just you'll never stop learning if you're truly fascinated in learning biodiversity.
Well, that's a great way to wrap it up. I'd like to thank everybody for coming. We had a huge audience, and most of them stuck around to the end, and there's a bunch more questions for you, Scott. But thank you, everybody, and thanks for a great seminar.
Thank you, everyone.